Today we will start our biology class for XII-C. Topic is fertilization. You know that what is fertilization? Fusion of male and female gametes. But how it is take place? First of all, germination of pollen grain and growth of pollen tube. See this. When the pollen is shed at the two-cell condition. Two-cell condition means when generative cell and vegetative cells are present inside the pollen grain, and the generative cell divides and form a two male gametes. And during the growth of pollen tube in the stigma, okay, this is the pollen grain and the pollen tube germinate, and this tube enters through the stigma and then towards the style. At this time, two male gametes are there, okay, and in the plant which shed pollen in the three-cell condition, pollen tube carry two male gametes from the beginning, and once the pollen has landed on the receptive stigma, the germination starts at the surface of the stigma, and the pollen, first of all, pollen hydrated by the absorption of water or the sticky or sugary substance which are secreted at the surface of the stigma. And after absorption of this sugary substance or water, this produce pollen tube after germination. And this stigmatic fluid secreted by the stigma contains sugar, lipid and resin, and thus produce a suitable medium for the germination of pollen grain. Same as the seed, after absorption of water, germinate. Same pollen grain after absorption of the stigmatic fluid which is secreted by the stigma are swelled by this medium and germinate into a pollen tube, and this pollen tube penetrates into the stigma by the activity of enzyme, and this enzyme is called cutaneous. And this, because of this, present in the cutine of the stigma at the place of contact with the pollen tube, the entry enters the entire contents of the pollen will goes towards the style, from the stigma towards the style, and germination move into the pollen tube, and growth of the pollen is restricted to the tip where most of the cytoplasm is concentrated. This emerging of pollen tube and penetrate from the stigma towards the style, this and then enters the ovule, and this channel take place by three methods. Because of the location or the place where this tube enters, these are divided into three parts. First, if entry of pollen tube into ovule is through micropyle, see, this is the micropylar region, this is the integument, this is the integument, this is the micropyle, and opposite to the micropyle, chalza and hair. So according to the location, the entry of the pollen tube is divided into three categories. First, progamy. Progamy means when the pollen tube enters the ovule through micropylar region, and this is called progamy. This is the most common example of the entry of the pollen tube into the ovule. Now second is chalzogamy. See the name, chalzogamy. Chalzogamy, when enters through the chalza end, when it's called pollen tube enters through the chalza end, it's called chalzogamy, and when enters through the integument, it is called mesogamy. Okay, according to the location, these are the three types. Now see, these are the examples. Chalzogamy, this is the common one. Progamy, common method. Chalzogamy, bitula, and mesogamy, cucurbita, and you have to learn this example also in each of the definition. Now entry of pollen tube in the embryo sac. This pollen tube into the ovule, it always enter into the embryo sac from the micropylar region. But see, synergized eggs are here now. This is the antipodal end and this is the egg apparatus. In this condition, center one is egg cell and two are adjacent are synergized. And this, when the micropyle or a progamy starts, the pollen tube enters between the egg and the synergized, or between the wall and the synergized, or between the two synergized. These are entry of pollen tube in the embryo sac. The synergized does not only play an important role in the determining the entry of the pollen tube. Okay. Now discharge of male gametes from pollen tube. After entering into the stigma through the style and into the ovule, the tip of the pollen tube burst and the two male gametes are discharged from this pollen tube. And the tube nucleus, two male gametes and one tube nucleus are there, but only tube nucleus disorganizes before bursting of the pollen tube. Only tube nucleus disorganizes, but male gametes remain here in the pollen tube. As these released by the help of amoeboid movement, these two male gametes inside this male pollen tube move towards the ovule, and one of them move towards the pollen nuclei and one towards the egg. And this two male gametes, one fuse with the pollen nuclei which form the endosperm, and the one male gamete fuse with the egg cell and this form the zygote. And this zygote, when zygote is formed or the fusion of male and female gamete, is called fertilization and diploid zygote form. But when male gamete fuse with this center one polar nuclei, this polar nuclei, then this is called triple fusion, because two polar nuclei and one male gamete, and that's what this three nuclei are formed, and fusion is called triple fusion. And the two male gametes fuse, that's why it is called double fertilization. Now double fertilization, you already know this. When fertilization involves fusion of one male nucleus with the egg, and when pollen nuclei with the pollen nuclei, it is called double fertilization. Now next, for post-fertilization. After fertilization, post-fertilization take place, and the post-fertilization events involve for events of endosperm, embryo development and maturation of ovule into seeds and ovary into fruits, and this development or the events involved is collectively termed as post-fertilization event. Now, endosperm development precedes embryo development. Why? Because first endosperm form and then embryo is formed. We see this, between this, the contact between one of the male gametes and the egg established earlier. See this example. The contact between the one male of the gametes and the egg. This, okay, male gamete and egg, this established earlier than the center one. Okay, but what happened? Primary endosperm is formed earlier. Even then, this is earlier fertilization, and then the second one is later. But formation, second center one form first, then the zygote. Why? Because cytoplasm of the central cell is more active than this egg apparatus region. That's why the center one portion active earlier than this. Okay. That's why I am explaining this first. Endosperm development precedes embryo development. The primary endosperm nucleus undergoes a series of division and form a ultimate endosperm. And this endosperm, which is formed at the center, this is very highly nutritive in function and provide nourishment to the developing embryo. Center is called endosperm, and this egg area, this form the zygote or the embryo. Now, this endosperm form, they are of three types according to the development, according to the divisions, according to the nuclear division, according to the cell wall formation or a free nuclei. These types. Endosperm are of three types. Okay. Now, types of endosperm formation. First is nuclear endosperm. Second type is called cellular endosperm, and third is halobial endosperm. Firstly, nuclear endosperm. Nuclear endosperm means when only nucleus divide without formation of cell wall or the free nuclei are present. Example, coconut. Inside the coconut, liquid sensation are present. How this liquid present inside the coconut? Because of the free nuclei, without the formation of cell wall, what happened? The nucleus move from one place to another because the nuclei are free in condition without any cell wall formation. That's why this is called nuclear endosperm. And this is the example of coconut water, and this coconut water, which is very nutritive material and helpful in culture of many experiments, and this is used as a nutritive medium in culture of experiment. It's also called as coconut milk factor, and this liquid is called liquid sensation. Now second is called cellular endosperm. In this type, cellular word, okay. In this case, the type of endosperm formation commonly occur in gamopetalous. You know gamopetalous? Gamo means fuse condition, fuse petals are there in dicot plants. In this case, the development is cellular endosperm. And this formation commences with the first division of primary endosperm nucleus. See, first nucleus, two nucleus divide into two, and then cell wall formation. But usually the cell wall are transversely formed, and this is of irregular size. Okay, and the first, which is near the micropyle region, is larger than the second chamber. Now see, in this case, in a cellular endosperm, this nucleus, if the nucleus are formed, the cell wall is formed in a regular pattern, but in halobial endosperm, this cell wall is irregular. This is the only difference between cellular endosperm and halobial endosperm. Cellular endosperm, cell wall formation by the first division of the primary endospermic nucleus, and first wall is usually laid down transversely, but subsequently division are irregular. First is normal, but subsequent divisions are irregular. And this results in the formation of endosperm tissue where cells do not show any regular arrangement. But in halobial endosperm, this type of endosperm, this is intermediate condition. First is nuclear, only free nuclei are there, no cell formation. In cellular, first of all, cell divisions are proper. First nuclear division, then cell wall formation, first transfer division, and then irregular. But in halobial endosperm, this is intermediate between the nuclear and cellular type. Okay? And the first division of primary endosperm nucleus is accompanied by the formation of transverse wall, and this divide embryocyte unequally. By the first division, irregularity start. But in cellular, first regular and then irregular, But in halobial, first divisions is irregular. Chalza 1 is micropylar 1 is larger and the Chalza chamber smaller. Okay, and then later on all the division take place, and this division of the embryos are unequal into a small Chalza chamber and the large micropylar chamber, and now the followed by free nuclear division. See, this is the nuclear type. First is cellular type and second is nuclear type. So this condition is called intermediate between nuclear and cellular type. Okay, but there are relatively few divisions in the Chalza chamber and this region often degenerative. Okay, and the free nuclear division of the micropyle chamber are followed by a wall formation and the cellular endosperm tissues are formed. So halobial is the intermediate, cellular one cell division, proper cell division, and the nuclear one only free nuclei without any cell formation.